Title of my co-op project is Type 1 Collagen-Targeted PET Probe for Fibrosis Detection in Mouse Models of Pancreatic Cancer. Abnormalities in the extracellular matrix is a hallmark of many different types of cancers. And one of the processes that we see occurring abnormally in this extracellular matrix is abnormal fibrosis, which can also be characterized by abnormal collagen distribution and excessive collagen deposition in the tumor microenvironment. Fibrosis is common in many solid tumors, including pancreatic ductal adenocarcinoma, also known as PDAC. You also see increased fibrosis in tissues treated with chemotherapy in PDAC and other types of tumors as well. Currently, there are limited techniques for non-invasively being able to characterize fibrosis in collagen, and we still need better and faster methods of monitoring treatment progression. Therefore, the goal of our project was to evaluate a novel PET probe for detection and quantification of fibrosis in pancreatic cancer for 2 mouse models of PDAC and monitor their treatment progression using this probe. The PET probe that we used for our imaging was CBP8, or collagen-binding peptide 8. This peptide binds specifically to newly formed mature type 1 collagen. We have previously used this probe in detecting fibrosis and monitoring treatment response in pulmonary lung fibrosis models, but this is the first time that we're now using this probe for PET imaging of cancer. For our PET imaging, we labeled with a radioisotope, gallium-68. This radioisotope has a very short half-life, which means reduced exposure to radioactivity for patients, and is also relatively easy to work with compared to some other traditional radioisotopes for PET imaging. For this project, we began by growing our cells in culture. We started with PDAC6 and PANC1, the 2 models that we used. These are both human pancreatic cancer cell lines. We implanted these cells in mice and allowed their tumors to grow. And once they had grown to a sufficient size, we imaged these mice before beginning treatment with our PET-MR machine using the CBP8 probe. We then began treatment with FOLFIRINOX, which is a chemotherapy drug cocktail that's used in humans for advanced pancreatic cancer. We administered dose every 3 days, and then we imaged the mice again after the 3rd dose and after the 5th dose, or about 1 week and 2 weeks respectively after beginning treatment. We also, throughout this process, collected tumors from these mice for ex vivo analysis, including immunohistochemistry and HPLC for hydroxyproline. While the study is still ongoing, I learned a variety of lab techniques and skills, including animal work, cell culture, immunohistochemistry, immunofluorescent staining, HPLC, Western blots, and PET-MR imaging. I have examples from some of the results of our study. The first is imaging results from a PET-MR imaging, where we can see uptake around the tumor, which is circled in yellow, of our specific probe, CBP8, which targets collagen. And then we also have immunohistochemistry stains specific for collagen-1, which is stained in brown from our tumors. And we can see that there is collagen deposition in these tumors. I also learned many research skills, including the field of molecular imaging for cancer and other disease types. I'm learning about the process of planning a research project and problem solving, as well as learning about the broad scope of cancer-related research, both in diagnosis, treatment, and in pathogenesis. In the future, in the short term, I plan to continue working in the Caravan lab on this project and other projects for the duration of the summer and potentially part-time during this academic school year. And long term, this CaNCURE co-op has reaffirmed my interest in research, and I plan to pursue graduate studies in molecular biology, hopefully focused on new diagnostic and treatment tools for cancer and potentially for other diseases as well. Lastly, I would like to acknowledge my faculty mentor, Shadi, my PI, Peter Caravan, the members of the Caravan Lab Group, and the Northeastern CaNCURE faculty for their support and guidance throughout this experience. Thank you.